Welcome to your personalized deep dive. Looks like you're fascinated by gene therapies for neurogenetic disorders. You've sent over some really interesting articles and research.
Definitely seems like you're interested in understanding the landscape, how tricky it is to treat the brain, what kinds of therapies are out there,
Yeah,
and the role of those patient advocacy groups. It's amazing what they're doing.
yeah, it really is. And one thing that really jumped out at me is just how many neurogenetic disorders are out there. Like the
Mhm.
research mentions over 1,700
Oh,
genes
yeah.
linked to these conditions. It's a
Yeah,
crazy number.
it really is. And each disorder can be caused by so many different types of mutations.
Oh, wow.
It's not just one thing. It's not like, oh, this one gene is always the problem. You could have like a tiny typo in the DNA code, or
Right.
you could have like a whole chunk of DNA missing or duplicated or even like shuffled around.
Wow, that's wild. I hadn't thought about it like that.
Mm.
And then on top of all that, you've got to actually get any treatment past the blood-brain barrier,
What?
which is like notoriously difficult.
It is. It's a major challenge. It's like, you're trying to get into Fort Knox.
Yeah,
Us-
exactly. It's
Yeah.
really protective.
So let's talk about some of the therapies then.
Okay.
Um, the research mentions ASOs, gene replacement therapy, RNA interference.
It's like a whole toolkit,
It
right?
really is.
Yeah.
Yeah.
So maybe we start with those ASOs,
Yep.
antisense oligonucleotides. It's mouthful, but
Yeah.
basically they're these short little strands of DNA or RNA, and they're designed to like target a specific RNA molecule in the cell
Okay.
and kind of mess with it, like change how it behaves.
Okay. And I remember reading that ASOs are already being used to
Mm.
treat some neurogenetic disorders.
Yeah, yeah. So there's nusinersen, which is approved for SMA spinal muscular atrophy,
Oh, right.
and that was a really big deal because it was the first ASO approved for a neurological disease.
Wow.
So that was kind of a groundbreaking moment.
That's amazing.
Yeah. So, um, but it sounds like ASOs aren't the only approach. What about this gene replacement therapy?
Right. So with that one, the idea is basically to deliver a healthy copy of the gene.
Oh, okay.
Right. So if there's a faulty gene, you're just giving the cell a good copy and like hoping it'll take over.
Like replacing a broken part?
Exactly. Yeah.
Yeah, okay.
Yeah.
So how do you get that healthy gene into the cells?
Well, that's where things get really interesting. They often use these like modified viruses.
Viruses, hold on.
Yeah.
Those are bad.
I know. But they're not the kind that'll make you sick.
Okay.
These are like AAVs, adeno-associated viruses. And basically, scientists have figured out how to take out the harmful parts and use them as like tiny little delivery trucks
Okay.
to carry the therapeutic gene into the cells.
So it's like they're sneaking
Exactly, like tiny Trojan horses.
good genes in. Yeah, okay.
And what's really cool is they can engineer these AAVs to target specific types of cells in the brain too. So it's not just like going everywhere.
That's incredible. That makes a lot of sense.
Yeah.
I mean, you'd want to make sure it's going to the right place.
Right?
And safety would be like a huge concern, I imagine.
Oh, absolutely. Absolutely. Safety is always like the top priority.
Yeah.
And they do a ton of testing to make sure these things are safe and effective before they ever get anywhere near a patient.
Okay, that makes sense. So we've got ASOs, we've got gene replacement. There's also this RNA interference that I was reading about.
Yeah. Oh, RNAi. So this one uses these little molecules called siRNAs,
Okay.
and they basically bind to a specific RNA molecule and prevent it from like being turned into a protein.
Oh, okay. So it's like
So it's like silencing the instructions.
turning off the production of a protein.
Exactly.
Yeah.
Yep. And
Yeah.
and they're really using this for things like Huntington's disease. Like there are clinical trials going on right now for that.
Wow, that's amazing. So many different ways to approach this. And I
Yeah.
also saw tRNA therapy mentioned in here.
Oh yeah, that's a really interesting one.
That's a whole other.
That's where we'll pick up next time.
Okay. I'm excited to learn more about that one.
Yeah, it's pretty cool stuff. So we were talking about tRNA therapies before the
Yeah.
break. tRNA therapies.
Those sounds super interesting.
Yeah, they really are. But to kind of get how they work, we need to go back to like protein synthesis 101.
Okay.
You remember, like, DNA has the code, but it's RNA that actually carries the instructions to the ribosomes where proteins are built.
Right. Yeah. DNA is the blueprint. RNA is the messenger.
Perfect. Exactly. And tRNAs, transfer RNAs, They're like the interpreters. They bring the right amino acids, the building blocks of proteins, to the ribosomes based on what the RNA tells them to do.
Okay, so if there's a mistake in those RNA instructions,
Right.
the wrong amino acid could get added, and then you've got a messed up protein.
Exactly. And that's where these tRNA therapies come in. They can kind of fix those errors.
How?
So one way is with what they call suppressor tRNAs. Yeah.
Okay.
And those are designed to like override those premature stop codons.
Oh, right, the stop signs that can make the protein too short.
Exactly.
Okay.
So the suppressor tRNAs can just ignore the stop signs and let the protein keep being built.
Oh, that's cool. So it's like telling the ribosome, nope, keep going, don't stop yet.
Exactly. That's a great way to put it. And what's really cool is that a single suppressor tRNA therapy could potentially treat a bunch of different disorders as long as they're all caused by that same kind of stop sign error.
Oh, that's amazing, like a universal fix.
Yeah, exactly. And then there's another type of tRNA therapy that's all about optimizing codons. So codons, you remember, those are the 3-letter sequences in our DNA and RNA.
Yeah, each codon's like a word in the genetic language.
Right. And it turns out some codons are just more efficient than others.
Really?
Yeah. Some lead to faster and more accurate protein production.
Huh. So how do the tRNA therapies help with that?
Well, they can engineer the tRNAs to recognize those optimized codons and make sure that the whole protein synthesis machinery just runs more smoothly and produces more of the protein we want.
Oh, cool. Like fine-tuning the engine.
Exactly.
Right.
Exactly.
That's so cool.
And there's even research on tRNAs that can actually extend the lifespan of messenger RNA, you know, the molecule that carries the instructions from DNA to the ribosomes.
Oh, so it's like giving those instruction carriers a longer shelf life?
Exactly.
So they can make more protein.
Yep. So we've got suppressor tRNAs overriding stop signs, codon optimizing tRNAs boosting production, and even some that can extend the lifespan of messenger RNA. It's a pretty exciting field with a ton of potential.
It sounds pretty amazing.
It is.
But I imagine there are still challenges, like with any kind of new therapy.
Oh, for sure. One of the big ones is delivery. You know, just like with those other gene therapies, getting the tRNAs past the blood-brain barrier and into the right cells.
Right. That barrier again.
It's a big hurdle. And then safety is always a concern too, obviously. Anytime you're messing with the genetic code, you want to make sure you're not going to cause any unintended consequences.
Yeah, of course. But it's still really exciting to see all this progress. I feel like we're really on the verge of some big breakthroughs.
We are. It really feels like it. And researchers are looking at even more stuff beyond tRNA therapies, too.
Yeah.
Like there's all this buzz around gene editing with CRISPR.
CRISPR, yeah, I've heard that term, but I don't really get how it works.
So CRISPR is it's a gene editing tool that lets scientists make very precise changes to DNA sequences. Like it's like a molecular scalpel that can cut and paste genetic code.
Oh, wow. So instead of just like masking or working around those genetic errors, CRISPR could actually like fix them at the source.
Exactly. It could permanently correct them, which is incredible.
That's amazing. It seems like that could really be a game changer for so many of these diseases. What are some of the like challenges with CRISPR?
Well, delivery is still a big one. You've got to get it to the right cells in the brain without messing with other parts of the genome.
Right. You don't want to accidentally edit something you're not supposed to.
Exactly. And another challenge is that the CRISPR system itself is pretty big, which can make it hard to package and deliver using those viral vectors we were talking about, like AAVs.
So are there other ways to deliver it? Like I read something about these lipid nanoparticles.
Yeah. Those are basically tiny little spheres of fat molecules that can like encapsulate the genetic material and deliver it. They seem like they could be safer and more versatile than viral vectors.
That's great. It sounds like there's so much happening in this field, so many new ideas and solutions coming out all the time.
Yeah, it's a really exciting time to be following it. And it's not just about the therapies themselves. Remember, we were talking about how important those patient groups are.
Oh yeah, those research foundations are really making a difference. They're advocating, funding, even developing therapies themselves.
They are. They're doing so much to raise awareness, speed up the research, and just really help get these therapies to patients faster. It's amazing to see.
Yeah, they're really inspiring.
They are.
So, besides funding, what are some of the other things those patient groups are doing to help?
Well, they're really big on trial readiness.
Trial readiness, making sure everything's in place for those clinical trials to be successful.
Exactly. So they help identify and recruit patients for the trials.
Okay.
Make sure the patients understand what's involved and provide support throughout the whole process.
Wow, that's incredible. It's like they're creating this bridge between the researchers and the patients.
Exactly. And they also work to make sure the trials are designed with the patient's needs in mind.
Okay.
Like they advocate for endpoints, the ways they measure if a therapy is working that are really relevant to how the patients are actually doing in their lives.
Right. So it's not just about whether it works in the lab, but whether it actually makes a difference for the people taking it.
Exactly. And they're also pushing for therapies for ultra-rare disorders, the ones that might not be profitable enough for big companies to bother with.
Yeah, that's so important.
It is. And because of all the work these patient groups are doing, more and more of those ultra-rare disorders are getting the attention and research they need.
So we've got this whole toolbox of gene therapies coming up.
Yeah.
ASOs, gene replacement, RNAi, tRNA therapies, even CRISPR. And we've got these amazing patient groups fighting to make it all happen. It's really incredible.
It is. It's an exciting time to be following this field. We're just at the beginning of what's possible. But there are things we need to think about, too. Like safety, how well these therapies actually work, and all the rules and regulations around them.
Yeah, those are all really important points. It sounds like there's a lot to consider as these therapies get closer to being available to patients. What happens next? It really does feel like we're like on the edge of a whole new world in medicine with all these gene therapies in development. But yeah, you're right. We need to think about more than just the science part of it.
Absolutely. As these therapies get closer and closer to actually being used to treat people, there are all these questions that come up about safety, of course, and how well they work, but also like the ethical stuff and what this all means for society.
Yeah, you mentioned the regulatory landscape earlier. What are some of the challenges there?
Well, gene therapies are still pretty new
Yeah.
and they're complicated. So the regulators are still figuring out like what's the best way to evaluate them? How do we know if they're safe and effective? It's not like approving a regular drug where we have like tons of experience and really clear rules.
Yeah, that makes sense. So how do they approach it then
Yeah.
with these brand new therapies?
It's tough. One of the biggest challenges is like how do you make sure patients are safe but also not hold back progress by being too cautious?
Right. You don't want to stifle innovation.
Right.
You also don't want to put people at risk.
Exactly. So it's a real balancing act.
So what kinds of things are they looking at specifically when they're evaluating a gene therapy?
Well, they look at a lot of things. How serious is the disease? What are the potential benefits of the therapy? But also the risks. And, of course, the data from all the research that's been done, like how good is the evidence that this therapy actually works? And then they look at the whole manufacturing process, too.
It's a lot to consider.
It is. It's a super rigorous process.
Yeah, and I guess it should be. We're talking about changing someone's genes, right? Like, that's a big deal.
It is a big deal. It's not something to take lightly. It could have really long-lasting effects.
Yeah, and another thing I was thinking about is the cost of these therapies.
Yeah.
I've heard gene therapies can be super expensive.
Yeah, that's a really important point. And it's something we need to figure out as a society. I mean, gene therapies are often personalized, right? They're made specifically for each individual patient. And that means the manufacturing process is really complex, and that drives up the cost.
So how do we make sure that everyone who needs these therapies can get them?
Yeah.
Not just the people who can afford it.
Right. That's a huge question, and people are talking about it a lot. We need to find new ways to pay for these therapies and make sure insurance will cover them. Maybe spread the cost out over many years, or figure out how to make manufacturing cheaper.
It's a matter of fairness, isn't it?
Mm.
Everyone deserves a chance to benefit from these advances.
Absolutely. And then there's the whole ethical side of things, too, especially with gene editing, like CRISPR. These tools could permanently change our DNA, and that raises some big questions about what it means to be human and how far we should go with all this.
Yeah, I can see how that would be a concern. What are some of the ethical dilemmas that people are talking about?
Well, one of the big ones is, like, where do we draw the line? Should we only be using gene editing to treat serious diseases? Or could we use it to, like, enhance people, make them stronger or smarter or whatever? And what about using it to choose what kinds of traits our kids will have? These are really tough questions.
Yeah.
And there are no easy answers. It's something we need to talk about as a society. Scientists, ethicists, policymakers, everybody.
It sounds like we're entering a whole new world with all this. It's kind of scary, but also really exciting.
It is. It's both. It's like we have this incredible power to help people and make their lives better in ways we never could before. But we have to be really careful. We need to make sure we're using this power responsibly.
This whole deep dive has been amazing. It's given me so much to think about. We're on the verge of something really revolutionary with these gene therapies. But we need to be aware of the challenges, too. and the ethics of it all. It's a lot.
It is, and this is just the beginning of the conversation, really.
Yeah.
The more we learn about gene therapies and gene editing, the more questions are going to come up. It's going to be an ongoing process.
So for everyone listening out there, stay informed, ask questions, and be part of this conversation. It's a journey we're all on together, and it could lead to an amazing future for human health.